So the question is, what are the gaps of knowledge and understanding multiple sclerosis? In terms of relapsing remitting disease, one never really knows causality. You don't know what you don't know. But I would argue that we have a very good working model of relapsing remitting MS in terms of the genetics, the basic immunology, the mouse models, but most importantly the clinical trials, which have in fact confirmed what all the basic science has indicated causes MS, which is that it's an autoimmune disease, autoreactive T cells against mild antigens get activated, traffic into the central nervous system, and cause relapsing remitting MS. So I think we understand that, and we have good ways of treating it. The big gap is in progressive MS, or secondary progressive MS. Here, blocking T cell traffic into the nervous system doesn't really help. The therapies are relatively ineffective compared to relapsing remitting MS. And we really don't have the same type of grasp or handle as to what is happening mechanistically in what is clearly a central nervous system disease as opposed to a peripheral immune disease with relapsing MS. So I think that is the biggest gap in our understanding of MS. Well, so how has all the genetic investigations led to our understanding of multiple sclerosis? I think it's given tremendous insight into the underlying cause of MS. We now have 2 papers recently published, one in the journal Cell, and another that has just been accepted in the journal Science. We've identified 233 common genetic variants and 8 rare genetic variants, which explain probably about half of the genetic architecture of MS. What has it taught us? The beauty of the genetics, let me just say, is there's no bias. There's no hypothesis, other than the fact we know that MS has a strong genetic component. So we can ask without a previous hypothesis as to, is this an immune disease or is this a neurologic disease? And the answer is incredibly clear. The genetic variants in MS are mapping to immune genes, not to nervous system genes. We, with our colleagues at the Broad Institute, with Brad Bernstein, published a paper in Nature about 3 years ago showing the variants in MS very specifically linked to immune cells, and this was replicated in a recent paper from the International MS Genetic Consortium, which are coming out. So what genetics has taught us is underlying pathophysiology of disease. It's identified the NF-kappaB pathway as being important. What the genetics has not taught us or been useful for is clinically. We've not been able to use the genetics to add that much more knowledge to predicting who will develop MS. It adds a bit, but not enough to be useful clinically. But that was with the previous work. So we'll now see with the new genetics if we can increase the predictability of whether someone will develop MS based on the genetics. Perhaps one of the biggest breakthroughs in medical science is the ability to do single-cell sequencing of cells. Because previously we took bulks of cells, we looked at a few proteins on their surface, but single-cell RNA sequencing allows us to take a tissue like spinal fluid or brain and blood and to sequence every single T cell very, very deeply to understand the nature of a disease. And we're doing it without a preconceived hypothesis. And this is an absolute game changer. So we've been applying this to MS. I'll tell you it's a very recent observation, which is about ready to submit for publication, which I think for myself represents the most important work we've done in my laboratory. So we've been sequencing cells from the blood and spinal fluid of patients with multiple sclerosis. And not unexpectedly, the spinal fluid in MS is very inflamed with a very strong signature of a particular cytokine called gamma interferon, which is thought to be an inflammatory cytokine. But they would then start to ask to look at normal controls. That is, we did spinal taps with proper IRB, and I don't know who's being tapped, and the graduate student arranged this. So we've done a series of spinal taps on healthy young individuals. We cannot tell them apart from MS. It turns out that the inflammatory signature we've been seeing, that I've been looking at for 40 years in MS, is really that of normal spinal fluid. And we see the same signature in the brain. And what's happening is the T cells, the subpopulation of T cells, migrate into the central nervous system. They're exposed to astrocytes, the glial cells in the brain, and they drive them to make gamma interferon. We found that cholesterol, a constituent of astrocytes, drive this inflammatory response. And we know from other literature in mice that if you knock out gamma interferon, they develop depression, anxiety disorders. And we didn't know where the gamma interferon was coming from. So we now know what's happening is that the astrocytes are driving the T cells for this neurodevelopmental function in the central nervous system. So what makes MS different? Well, we found that what's different is not the general milieu of T cells there, but the antigen-specific cells. There we see orders of magnitude difference between MS patients and control. The MS patients have high frequencies of mild and reactive T cells that we can directly identify, which are absent in healthy individuals. So this technique has just given us new insight in unexpected ways. So the question is, what are the environmental triggers leading to the dysfunction of immune cells in MS? So a few years ago, we discovered that salt, high concentrations of salt, drove inflammation in normal T cells and in experimental models. If you fed a high salt diet to mice with the experimental model of MS, EAE, the EAE was much worse. We then went on to show that the salt led to dysfunction of regulatory T cells. We also know that fat leads to dysfunction of regulatory T cells. But that's basic science, and what we don't know is how that really translates to the human disease. The epidemiologic studies have been mixed. Some have shown the effect of salt, some others have not. So I think it's safe to say that we shall eat well. We should have low-fat, low-salt diets. We should exercise every day. Whether that actually changes the outcome of patients with MS is, I think, difficult to say, but I still advise my patients to eat a healthy diet. So what is the role of NF-kappaB and other transcriptional targets in driving MS? When we looked at the genetics, about 18% of the genetic hits are in the NF-kappaB pathway. NF-kappaB is sort of the mother of all transcription factors for inflammation. Was the first transcription factor discovered decades ago. When we map the genes as to where the genetic variants are hitting transcription factor binding sites, they hit in NF-kappaB. So we think NF-kappaB is important, and we know that in MS there's an increased NF-kappaB signal. So we are curious as to how that is related to MS. Some very preliminary data by, again, single-cell RNA sequencing after treatment with B-cell depletion, we're finding that the NF-kappaB pathway is very much altered after treatment. So rather than specifically targeting this pathway, we believe that a number of the effective treatments will in fact be targeting NF-kappaB somewhat as a measure of inflammation.